Olá, pessoal! Nós sabemos que 2020 tem sido um ano difícil e que o distanciamento social proporcionado pela pandemia Covid-19 pode nos deixar um pouco desanimados.
Bem, não poderíamos deixar de prestigiar um evento tão aguardado e importante para a nossa comunidade científica, que este ano completa sua 10ª edição.
Então é com imenso prazer que anunciamos a realização da 10ª edição da Semana da Química da Universidade de Sorocaba.
A semana foi pensada e organizada de modo que todas as atividades acontecerão online, seguindo todas as normas necessárias para garantir a segurança da equipe organizadora e de nossos colaboradores.
Esse evento tão aguardado ocorrerá nos dias 27, 28, 29 e 30 de outubro e nos dias 3, 4 e 5 de novembro. Lembrando que nesses dias haverá um recesso no semestre não presencial da UFSCar, então não há desculpa para você faltar nesse evento maravilhoso.
Nele teremos diversas novidades e grandes atrações, dentre eles parceiros, patrocinadores, sorteios de brindes e palestrantes de grande notoriedade.
E hoje temos um convidado de renome, o Dr. Jean-Pierre Sauvage, químico francês, professor da Universidade de Estrasburgo e ganhador do prêmio Nobel de Química de 2016, vai contar pra gente um pouco sobre a sua pesquisa. Bora conferir?
Let me thank the organizers of this very promising meeting, Semana da Química. I'm really looking forward to explaining what we have been doing in the last decades, and I hope you will enjoy my lecture. Thank you. The first publication on chemical topology appeared in 1961. It was a JAX paper. It's a pure discussion. There is strictly no experimental work reported in this paper, but I believe it is important. Historically, it's a very important paper. Just a quick example, if you look at the top line, you take a ring or a circle. It has a planar graph because no problem to draw it in a plane without crossings. Now you take exactly the same sequence of atoms and chemical bonds, and you can arrange the system in a different way. And this is represented on the right. You have a knotted ring, and this knotted ring has a non-planar graph because you must have 3 crossings if you want to draw it on your sheet of paper. So that was really the beginning of a new field. Now let's move on towards real chemistry. The catenane is a molecular system consisting of several interlocking rings. Such systems can be very simple, some others are much more complex, and the simplest one is a 2-catenane as represented on the slide. The 2-catenane consists of 2 interlocking rings. This is, of course, a molecular system which is topologically non-trivial because simply its graph is non-planar. You must have 2 crossings if you want to represent a 2-catenane in your sheet of paper. Catenanes have been considered by chemists for many, many years, and there were many discussions from the beginning of the 20th century regarding these molecules. So they are, of course, very exciting, topologically exciting, but also they may display interesting properties. And for years and years and years, they were nothing but discussion objects. Nobody could make them, and nobody could characterize them, so they appeared as very exciting, very attractive, but still mysterious. The first synthesis of a catenane was published in 1964 by 2 German chemists, Professor Schill and Professor Lüskinghaus. It was a beautiful piece of work based on traditional organic  Historically, it's also very important There was nevertheless a difficulty, which was that it was a 23-step synthesis, so very long, very tedious, and they probably couldn't make more than a few milligrams of the final molecule. Thus, some new strategies had to be discovered in order to make these catenanes accessible molecular systems. I will now explain you why we became interested in catenanes and more generally in chemical topology, and I will try to show you that starting your research in a given field and at some stage deciding that maybe you can jump quite far away into another field can be very beneficial. When I started my group in 1980, we were very much interested in water splitting, trying to split the water molecule to H2 and O2. It was also a time of solar energy research, which became very, very active. And so that seemed to be extremely attractive. At the time, the real hero of all inorganic photochemists was ruthenium trisbipyridine, which is drawn on the slide here. It's a very attractive compound, which is still used and extensively studied. Very, very stable, highly colored, so it had a lot of very promising properties in relation with electron transfer and finally water splitting to H2 and O2. The weak point of ruthenium trisbipyridine was and still is that ruthenium is a noble metal, very, very expensive, and so it was probably of interest at the time, and it is still today, to try and replace ruthenium by some other metal, a much cheaper metal. And I will explain you how we started to work on copper, trying to replace ruthenium for copper. At the beginning of the 80s, David Macmillan, an expert of copper photochemistry, spent his sabbatical leave in Strasbourg. We interacted very strongly from the beginning, and when he saw the molecule we had just made, which is on the left here, a diphenylphenanthroline, he became very excited because he had the feeling that this molecule could lead to a very interesting copper complex. We thus started to collaborate immediately, and we made the copper complex which is shown on the right. This copper complex turned out to be very interesting. It had very interesting properties in its excited state, with the ability to transfer electrons easily to other molecular systems. However, there was something else, probably more important, which drew my attention. If you consider the two endpoints of the vertical ligand, which are represented in blue, and the two red points of the horizontal ligand, represented in red, and now if you can connect the two blue dots and separately interlink the two red dots, you are in a very good position for making a catenane. Looking at the two rings now you have obtained, they are clearly interlocking with one another. So, with my good friend, Christian Dietrich Büchecker, a CNRS researcher, who was at the same time a fantastic organic chemist, we decided to embark in this project of making catenanes based on this very simple idea that copper complexes could be used as precursors. So copper will be the templating ion, and we will arrange organic molecules around the copper center so as to favor formation of a catenane. Christian was incredibly efficient, and within 10 months or so, we published our first paper, which is shown here on the slide. The first paper describes the synthesis of a catenane. It's an easy synthesis, very accessible. You can make large amounts of catenanes, grams if you like. And we decided to publish it in French, but I'm not going to insult you and translate it to English. It's pretty obvious, and we published in Tetrahedron Letters, which was a reasonably good journal, but not a high-impact journal. The reason was simply that we didn't know what high-impact journals were. It was not so important to the scientists at the time, and we just published in a reasonably good journal. That was it. The slide shows the two strategies which had been used in order to make a catenane. So relatively simple strategies. Strategy A, which we call entwining. You take a copper center, the black circle, you mix it with two organic fragments, which are FF fragments, F being the terminal functions. You generate an entwined species, and you cyclize using GG fragments. You make your catenane on the right in a double cyclization reaction. The second strategy is very, very similar, except that you have to pre-synthesize a ring from a FF fragment and a GG fragment. And we called this strategy the gathering and threading approach. As you will see in a few seconds, the video speaks by itself and doesn't need a long comment. We first proceed to the entwining reaction, and this is followed by the double cyclization reaction leading to the catenane. The second strategy requires the synthesis of a ring before proceeding to the gathering and threading reaction, and finally to the single cyclization reaction. Interestingly, one can easily remove the copper 1 center from the complex catenane. This is done very easily using potassium cyanide, and the reaction is quantitative. If we want to go back to the copper complex, one takes the free ligand represented on the right here, and we add copper 1. Very, very simply. Everything is quantitative, everything is very fast. We also had the X-ray structures done, and the X-ray structure of the copper 1 complex is shown on the left. Once the system has been demethylated, one obtains a completely different structure, which is the one on the right. At the very beginning of the work done by my group on catenanes, the yields were very modest. The overall yield for making the very first catenane, which we have seen before, was something around 25–30%. Later on, we improved very, very significantly the synthesis by using the ring-closing metathesis strategy, which was developed originally by Bob Grubbs. And in some collaborative work, we could show that the catenane can be made with an overall yield of 92%, so extremely high. The strategy is shown on the slide, going from 2 organic fragments on the left, plus copper 1, one obtains the entwine system, and by applying the ring-closing metathesis reaction, one can cyclize and obtain both rings. The system can even be demethylated, and if we want, it can be hydrogenated to the fully saturated catenane. Let me now pay homage to the real pioneers who contributed to the field of catenanes. At the very beginning, Ed Wasserman proposed a statistical approach, but more important, he published the very nice paper in JACS in 1961 explaining what chemical topology means. Professor Schill and Professor Lüttringhaus, as I already mentioned, also brought a very important contribution, fully based on organic synthesis. And later on, much later on actually, our group started to work in the field, followed by Fraser Stoddart and his co-workers, who published their first contribution in 1989. Chris Hunter and Fritz Völkle applied basically the same strategy based on hydrogen bonding to make catenanes, and they could do that very efficiently. They published in 1992. More recently, Professor Fujita in Tokyo reported a beautiful synthesis of a catenane based on palladium-nitrogen bonds, which are labile, thermodynamically stable, and they allow rings to be interlocked with one another by opening a bond and then reforming the bond. Those people were the real pioneers, and later on, the field basically blew up, and many, many outstanding research teams contributed to it. Let me mention the work of Professor Ogino, one of the real pioneers in the field of rotaxanes, which are always associated to catenanes in chemistry. And his approach was based on coordination chemistry, published at a very early stage. A 2-catenane is with no doubt the prototype of topologically non-trivial species. It is shown on the left here. But later on, many groups, including ours, were very interested in making and studying more complex topologies, such as a 3-catenane on the right of the upper line. Very challenging were the synthesis of the trefoil knot on the left, bottom line. And this trefoil knot could be made, in particular by our group, some time ago. More complex species have also been prepared by various people, including our team, Fraser Stoddart's team, and many, many other research teams interested in molecular topology. We will now talk about molecular machines. First, molecular machines in biology, and second, molecular machines in pure chemistry. If we look at molecules which have been made by chemists, as you know, there are many, many, many synthetic molecules which have been reported. These molecules are most of the time considered as static. They may move, of course, but more or less at random, and basically before the work on molecular machines done by several teams, these molecules were considered as motionless. By contrast, in biology, controlled molecular motion is very important. It is essential and omnipresent. So in biology, one can find linear motors, rotary motors, even walkers, molecules which are able to walk on a rail or on a road, systems undergoing contraction or extension, especially in muscles. So motor proteins, this is the family of these molecules, are everywhere. Just two examples which are particularly important. ATP synthase is a rotary motor, and this is of course an enzyme which is totally essential to life. It is operating in all living organisms on Earth because it's related to ATP, which is the vector of the biological fuel. So ATP synthase can make ATP from ADP and inorganic phosphate. It can also hydrolyze ATP to ADP and inorganic phosphate. We will also discuss very briefly another essential machine, which is kinesin. And kinesin is a walker responsible for the transport of big molecular systems in the cell. It walks on a rail, or even it runs because it's a very fast moving system. And we will see a video. Here is a nice video which you can find on the web quite easily. And it shows that ATP synthase is a rotary motor. There is the internal part of it with an axis which spins inside a big cluster of proteins, the red and green proteins, forming some kind of aggregate with a hollow part. The process is driven by proton transport across the membrane. And if you look carefully, you will see that there are small white dots which are crossing the membrane. And this is the energy which is needed for setting the system in motion. So very, very important. You can also look carefully and see that ATP, which is purple, is made out of ADP and inorganic phosphate, respectively yellow and blue particles, and those particles are consumed while ATP is liberated. The kinesin is the tiny thing which moves, which walks on the rail, so it's kind of a cylindrical rail or a microtubule, and the kinesin is attached to a huge ball here, a huge bag, if you like, and in this bag there are the organelles, the molecular systems, which have to be carried from one spot in the cell to another spot. And it seems to walk quietly, slowly, but in fact it is not at all the case. It walks very fast, and it is more or less as if a human being was running at 200 kilometers an hour. We will now talk about molecular machines in chemistry. As you will see, the molecular machines and motors made by chemists are relatively complex molecules, but nevertheless, they are extremely simple molecular systems compared to biological machines and motors. This is, of course, not surprising. Chemists have always been fascinated by biology, and in particular by molecular biology. One of the dreams of many chemists was trying to reproduce the functions or structures reminiscent of biological functions, and this is, of course, very ambitious. At the beginning of my lecture, I spoke about catenanes, and you may wonder, what is the relationship between catenanes or rhotaxanes and molecular machines or molecular motors? As you will see, catenanes and rhotaxanes are perfect models for elaborating molecular machines. Catenanes and rhotaxanes contain two or more fragments which are not covalently linked with one another. And this is a very important property. So you can easily imagine that a ring can glide along an axis, like on the top example on the slide. It can glide from a position on the left to a position on the right and go back to the left. And this is very, very similar to a linear motor, a little bit like a piston moving in a cylinder. Alternatively, an axis can spin inside a ring, or if you prefer, a ring can spin about the axis. Similarly, if you can set in motion one of the rings of the catenane, which is on the right, whereas the other ring is fixed, you are very close to a rotary motor. So rotary motors are very exciting, but of course, they seem to be difficult to make. The first molecular machine made by my group is a 2-catenane complex to a copper center. And we will play with the two oxidation states, copper 1 and copper 2, knowing that copper 1 likes to be 4 coordinated, whereas copper 2, which is a D9 electronic configuration metal, prefers to be 5 or 6 coordinated. This slide explains the principle of our pirouetting machine with one ring, which is a little bit more complex than the other. The ring on the left contains 2 coordination sites, a bidentate chelate and a tridentate chelating group. Now going from copper 1 to copper 2, one can trigger the motion simply because copper 1, as I said before, likes to be 4 coordinated and copper 2 prefers to be 5 coordinated. So the fragment, which is originally on the left, on the upper line, will start to interact with copper 2 and kick away the bidentate chelate, which is originally coordinated to the metal center. This takes a bit of time, minutes, as indicated on the right part of the slide. And after you have obtained your 5 coordinated copper 2 complex, you can go back by applying a redox potential, which is slightly negative, which will afford you with the copper 1 complex, still 5 coordinates, and within a couple of seconds the system spins again and you regenerate the starting form of the molecule. This process is probably easy to visualize on the slide. So the copper center is either brown for copper 1, this is roughly the color of the complex, and it is green for the copper 2 center, which is also the color of the complex. And by going from copper 1 to copper 2, by simply abstracting an electron, or going back from copper 2 to copper 1, one modifies the shape of the system very significantly, and at the same time one forces the lower ring to move from one situation to another situation. That was the first molecular machine we made in Strasbourg already some time ago. The catenane we have just seen before is very sluggish. It takes minutes and minutes and minutes to move, to go from one situation to another. For copper 2 going back to copper 1, it is faster. It takes seconds. But we thought the system is not fully satisfactory. And then we devoted a lot of time, a lot of work to improving the system and to designing and preparing molecular machines which were much faster to move. And here we have an example after maybe 12 years of studies. This system, which is now a rotaxane, can pirouette or spin within microseconds or milliseconds, and we call it the 5th generation molecular machine. I will not restrict my discussion to the systems made in Strasbourg by my group, and I would like to discuss very briefly another very early molecular machine which was named a molecular shuttle by the group of Fraser Stoddart. The principle is quite simple. It is explained on the slide. The ring can glide from a position on the left to a position on the right by sending a signal to the molecule, and then by sending another signal to the molecule, it will go back to its original position on the left. So this is, as I said before, similar to a linear motor. Here is the compound made by Fraser Stoddart and Ankel Kiefer with their two co-workers, Dr. Bissell and Dr. Cordova. So you have a blue ring here. It's blue on the slide, it is blue in the drawings, but it's not blue in reality. And this ring is very strongly electron deficient. It can move from a green station, so this is the vocabulary they have been using, a station. The green station is very electron rich, so you have some kind of charge transfer interaction between the donor, green, and the acceptor, blue. And if you abstract an electron from the green station, you make it a positively charged, and it is not a good electron donor anymore, of course. So the ring will glide to the right and prefer to interact with the red station. You can go back by injecting an electron back into the system so as to regenerate the neutral form of the green station. Ben Feringa and his group have done fantastic work in the field of molecules in motion. And in 1999, they were the very first people to describe the synthesis of a rotary motor, a real rotary motor with a perfect control of directionality, which was strictly impossible before. So now we have the system they made 20 years ago. You have a double bond here which plays a very important role. You probably know that if you irradiate a double bond, you generate an excited state which tends to rearrange. And this is a very nice trick for inducing a rotation motion. So the system contains 4 components. On the left upper line, you have the starting molecule. Then by shining light on it, UV light, you start a motion. It starts to rotate. And by waiting for some time or by heating a little bit, the system goes on its rotation process. One obtains the third form on the bottom line on the right. And then you shine light again, UV light. The system goes on. And after heating in the fourth process, from the left molecule on the bottom line, you go back to the starting form of the molecule. And this is really something fantastic. Here is a video made by Feringa and his university. I think it is very demonstrative, and it explains exactly what happens. You have 4 steps. A photochemical step, so you see a big flash of light. Then a thermal step, you see the symbol of heating device. The third step is again a photochemical step, big flash. And the fourth step, which will allow you to go back to the starting form of the molecule, is thermal. Again, you have the heating symbol which appears on the slide. About 20 years ago, we started to work on contractile and extensible molecular systems, very much related to molecular muscles. We thought that it was important that chemists show they are able to make relatively complex systems reminiscent of natural systems, and in particular, molecular systems which can be contracted or elongated at will. And this is what we will now discuss. As you probably know, the way biological muscles move, contract, or extend is nowadays very well understood. You do not contract springs like what you could imagine intuitively, but rather you have filaments which glide along one another. In particular, the thick filaments, which are indicated in blue, glide along the thin filaments consisting of actin, it's a polymer, and those actin-containing filaments are thin. And so if you contract your muscle, you see very easily that you have to glide filaments, the thin filaments, toward the center of the ensemble of filaments, which is represented on the slide. And of course, if you want to stretch your muscle, you do exactly the opposite. About 20 years ago, we embarked in a very ambitious project which was trying to mimic biological muscles, and in particular, use the principle of gliding filaments along one another. And so it took some time, of course, but we ended up with a molecule which is very schematically represented on the slide here. It's a rotaxane dimer. You can see the 2 stoppers. You can see the 2 rings, a dark blue ring and a pale blue ring, and the 2 filaments threading the rings. And now, what we could show is that if you send a chemical signal to the molecule, you will modify its shape, you will contract the molecule, and if you send another signal to the contracted molecule, you will stretch it, you will elongate it so as to obtain the original shape of the compound. And this piece of work has been followed by many very exciting discoveries by many groups related to artificial molecular muscles, and in particular to polymers, which can be stretched or contracted at will. Let me now discuss the very last example of molecular machine designed, made and studied in Strasbourg. This particular example is that of a molecular compressor. Very ambitious, as you will see, but we had really fun making it and setting the molecular system in motion. The general project is explained on the slide. So we wanted to make a 4-rotaxane, so thus consisting of 4 components, 2 dumbbells and 2 bis macrocycles. And this 4-rotaxane should be able to move under certain conditions so as to bring the 2 plates represented in green here, kind of square plates, to bring them to close proximity when we want to do that. So that would be simply to move plates and perhaps to be able to incorporate something in between those 2 plates and contract the fig which has been incorporated, namely G, the guest. So if we can do that, we can indeed claim that we made a compressor. Now the design of the real molecule. So the real molecule we wanted to make is shown on the slide. It looks very complex, and several people in my group told us, or told me, that the project was not realistic and that we would never succeed in making such a complex molecular system. But if you look, it is highly symmetrical. And if you have highly symmetrical molecules in view, they are certainly much easier to make than molecules without any remarkable symmetry. And this turned out to be true. So we have the 2 dumbbells, as you can easily recognize. We have the bis macrocycles in the middle of the molecule, and each bis macrocycle contains a zinc porphyrin. And those zinc porphyrins will be the plates which we will be able to move and to bring to close proximity or to keep far away from one another. So now what we had in mind was to incorporate something in between those 2 plates and then contract the something, the guest, and see whether we can contract it and finally probably expel it from the recognition site, which is provided by those 2 zinc porphyrins. The synthesis of the compound was a real tour de force. It was successful because we used the copper trick, copper being able to gather all the organic fragments around it and thus favor formation of the rotaxane. It was also successful thanks to the talent of the people who were doing the work. So finally, we could make more than 100 milligrams of the molecule, and this was by far enough for studying it in detail. The icing on the cake came with the X-ray structure. We were indeed very surprised to see that we could crystallize the compound and obtain a high-resolution structure. Small molecules containing 2 basic functions able to interact with both zinc ions could be inserted in between the porphyrinic plates. By removing the 4 copper ions, the 2 porphyritic plates get closer and closer to one another, and the general structure of the molecule completely collapses. The guest, complexed in between the 2 porphyritic plates, is thus compressed and finally released. The rigid and rectangular structure of the compound can be regenerated by simply adding copper to it. The complexing ability of the compound towards small molecules is thus completely regenerated, and this is done in a very easy way. To conclude on this part, the 4-rhotaxane can be completely flattened and the complex gas must be expelled, in turn by regenerating the rectangular and rigid shape of the compound. This being done by adding copper, one regenerates the ability of the molecule to complex small compounds in between the porphyritic plates. So this is simply starting from a compressor, one obtains a totally inactive form which can be regenerated. To conclude, before the emergence of the field molecular machines and molecular motors, synthetic molecules were mostly regarded as motionless objects. The introduction of molecular machines modified completely the view that chemists were having on their molecules, going from a still object to an object which can behave as a machine or as a motor, thus undergoing large amplitude motions in a strictly controlled fashion. Before thanking the people who did the work in my group, absolutely fantastic chemists, let me stress once more what I consider as very important for scientists. Novelty is very certainly number one, and novelty is obtained by interacting with other scientists, and it is also obtained by meeting people, having discussions with people, listening to what they say to you, explaining what you are doing yourself to them. Jumping from a field to another is also something important, which can be very, very beneficial. You shouldn't ask yourself whether you are good enough to jump and to tackle a new problem, just do it. Finally, I would like to insist on serendipity. Many very important discoveries have been made thanks to accidents, so keep it in mind when you observe something which is experimentally very surprising. Try to dig and understand why you observed it. The work I spoke about today was performed in a highly collective fashion by a team of professional researchers and university professors, PhD students and postdocs. And here is a list of people who worked with me for many years, for some of them. They are university members, or they are CNRS members. Extremely important contribution from these people. We have been working in several fields in my group, in particular on artificial photosynthesis. On this list, I just included the names of the people who worked in the field of catenanes, rhotaxanes, and molecular knots. many people, very talented people, and I'm very sorry to say that we have no time to name them all. Very sorry for that. Similarly, here is the list of people who worked on molecular machines and motors, which I call here catenanes and rotaxanes in action. Aude Livret was the very first to make a molecular machine, and I would like also to refer to the work of Maria Consuelo Jimenez, whom everybody used to call Cello. She was mostly responsible for the design and the synthesis of the muscle in association with Christian Dietrich Buchiquer. The work on the molecular compressor or adaptable receptor was mostly carried out by the people who are listed in the upper part of this slide. Let me stress that I was very lucky to work with highly motivated researchers. These people were at the same time extremely competent, and it was a very nice combination of professional scientists and younger scientists, which turned out to be particularly efficient. Finally, let me thank my university, as well as the CNRS, the French research agency, the European communities, and other institutions for their support. A special thanks to Professor Jean-Marie Lane, my former supervisor many years ago, and a very close friend today, Malcolm Green, who very sadly passed away. He was a fantastic organometallic chemist, and I learned a lot with him when I was a postdoc in his group in Oxford, in the UK. I also thank some teachers who had a very strong influence on me, my family, and my two co-laureates, Fraser Stoddart and Ben Feringa, who are very good friends. Finally, let me thank you very warmly for your attention. Thank you.
Agradecemos a participação de todos os parceiros representantes do nosso evento inesquecível e ressaltamos que pensamos em cada detalhe com todo o carinho do mundo, para que sua experiência seja a melhor e a mais proveitosa possível.
E não se esqueça de nos acompanhar nas redes sociais para ter acesso a mais conteúdos como este e consultar nossa programação. No Instagram em @sequiufiscar2020, no Facebook em /semaquiufiscar e também em nosso site Semana daqui em soroca.wixsite.com/sequiufiscar2020.